So there we go. So we will going to study, start the chapter called targeted genome editing. The topic itself is quite obvious, but you need to understand as a medical biotechnology student that from where this thing come into the minds of the scientific community. And if you can recall our earlier discussions and topics, especially the gene therapy, most of your questions in gene therapy, those questions were related to the troubles and the limitations of the techniques of gene therapy. Can you recall some of the limitations and the troubles associated with gene therapy? Shabash. Who?
Sir, the gene may get inserted in place of another gene and stop the protein production of that gene.
Okay, uncontrolled.
And maybe multiple copies.
Unguided. Okay, Shabash, good. Unguided, yes. Unguided insertion of the gene. Nobody knows where the external gene will going to land within the host genome. That's one point. The other point is that we might ended up in multiple insertions in different locations on the chromosomes. So that was always the big concern when you try to manipulate higher animals. I mean, that might not be the issue if you are trying to engineer bacteria, viruses, fungus, or insects, or some other animal. I mean, nobody cares about the other species. But this is really important in case when you want to manipulate the human genome. Because when you are dealing with the human genome, you should be absolutely sure about the procedure, about the mechanism, and about the specific location where you want to insert your external gene. That's very important. Otherwise, the world will never be able to feel comfortable with the concept of genome editing. So you cannot manipulate the genome, you cannot edit the genome if you don't have an expertise of controlling the mechanism. When I said controlling the mechanism, that is about that you should absolutely sure where that external fragment of DNA will going to be incorporated into the human genome. Once we have like control of that thing, we might be able to start using that procedure for therapy and of course for other purposes as well. But right now, the biggest ethical concern on the human genomic DNA manipulation is that all the techniques which we were using were not, I must say, completely understood. We cannot control all the mechanism of insertions of external DNA. So there is a need to find better ways. There is a need to create tools which help us and which help the scientists to send the external DNA at the place of their choice. And I can tell you it's not an easy task. I mean, sending an external gene, for example, this is supposed to be a genome. Genome is not that much smaller, but for example, if this is the genome, and you want to send an external piece of DNA, this is an external piece of DNA, and you want to send this DNA to that place, the place of your choice. That's what we call as targeted genome editing. And this procedure is executed with the help of molecular tools. And those molecular tools are not evolved in one go. Those molecular tools are evolved over the period of like 3 decades. They evolved over the period of like 3 decades. Different scientists, different research groups had contributed in developing those molecular tools which allows us and allows human beings to manipulate the genome at the place of their own choice. I'm sure you understand that why this topic has, is so much important, because it is actually the bottleneck. It is the one thing which is stopping researchers and scientists to manipulate the genome freely. Because until and unless the scientific community do not address this concern, the world will not going to accept any methodology and any procedure to disrupt or to edit the genome. Because no matter whether you are trying to the stuff for the good, even you are trying to make editing for the better purpose, for a greater cause, if you do not have a control on the mechanism, that means it might lead to another problem. And the bad thing is that, the worst thing is that we don't know about the other problem. We don't know where it is going. So that's very special. I hope this helps you to understand why there is a need of targeted genome editing is out there. In order to understand the context of the genome, just try to relate certain interesting analogies. For example, in linguistics, we have alphabets. In English and in Urdu, we have alphabets. These alphabets, when they join, we put them together, they create words. And when these words were put together, they make sentences, paragraphs, books, libraries. So that is how we able to collect information with the help of the alphabets. The more advanced and sophisticated version of collecting information or organizing information is the binary code system. And most of you guys understand, who understand computers, that the basic information in the computer software is written in the form of yes, no, yes, no, yes, no. And that is called as the binary code. With just two alphabets, we can create trillions of types of combinations to generate several 100,000 types of instructions and information. But all the complicated information the operating system is carrying is actually arises out of this binary code. There's two alphabets. Think about here in the linguistic. I don't remember the number of alphabets in English, maybe 26 or 24. Agar main galat hua to please sorry for that. That many alphabets creating books. Think about just two alphabets creating the operating system. All the wonderful things we are having right now based on computers. Now, if you go to the biological level, things are pretty similar, but they are much more complicated and advanced. You might be wondering, more advanced than computer? I can tell you several folds more advanced than computers. Even today, I'm not talking about the future. Maybe in the future we might be able to create like such powerful computers which can outclass human brain and human information. So in humans or in higher animals, the information is written in the form of 4 alphabets: A, C, T, and G. So you can see that the nature has used 4 alphabets, just 4 alphabets, to create what we call as the life. The code of life was written with the help of just 4 alphabets. These 4 alphabets arranged in different patterns, in different sequences, in different numbers, to generate different types of functions, to store information, to store different types of information. So the 4 alphabets, when arranged In one way, it can control the height of the humans. The same 4 alphabets, when organized in another way, they can control the eye color. The same 4 alphabets, when organized in a different other way, they can control the skin tone. And they can control the functions of all possible functions executed within life. So that information of the 4 alphabets is stored in the form of DNA, and the higher order level is what we call as genome. So genome is actually the collection of the information. It's actually the collection of information. It's actually like an uninstalled software which is carrying all the information. So now that information on the genome is present in the form of genes. And I told you on the other day that life is not about having the DNA. It's not about having the genes. It's about how we can regulate the expression of those genes. Which one will going to be expressed and which of the genes will not going to be expressed. This control is what we call as the life. But in a nutshell, I can tell you right now is that human genome is a collection of information which can control the functions of life, or which can control humans. Now that genome, human genome, is so big, is so large, that if you want to make changes at one place of the genome, I can tell you it is like throwing the stone from the International Space Station onto the earth, which hit on your head. It is like that, I can tell. I don't want somebody to hit your head with the stone. But imagine the difference and the difficulty in the approach of targeted genome editing. The genome is so big, and you want to make changes in one small place on the, it's like in the gigabase pairs out there. So gigabase pairs, 10 to the 9 base pairs out there, and you want to manipulate few base pairs out of it. It is that much complicated and difficult to go at the place of your choice, and then to edit the genome at the place of your own choice. That is why it is now currently considered as one of the most advanced and leading area of research of big scientific research groups out there. So we have developed a lot of advanced tools for that thing, and I will going to introduce them. If you have any question, please feel free to ask me, okay? Just for a comparison, just see that the size, the difference in the size of the genome. Human genome as compared to the bacterial genome. A bacterial genome is just 4.6 megabase pairs. And think about the human Homo sapiens genome, which is like 3.2 gigabase pair. And I can tell you, at that level, when a value is increased like 10 times, it is not like you are going from 1 to 10. Like from million to the 10 million, 10 million is too much. From the 10 million to the billion is too much. So every zero which will be added after that, that going to change the value exponentially. So we have a huge amount of data present in our genome. So that is actually the reason of complexity behind human beings. But that complexity is giving us some trouble, but that we are not very much comfortable in editing and finding the right place to edit it comfortably. Okay, and you know, changing human genome has their own consequences, because we are very much similar with the other species, very close to the other species. I can tell you They're close, approximately 98.4% similar with the primates, with chimpanzee. So there's just a difference of 1.4 to 1.6% in the genome of humans and the chimpanzee. So हम में से बहुत सारे लोग बंदर नहीं है, सिर्फ 1.6% के difference की वजह से. So there are different molecular tools are out there, and now you should be able to understand that why these molecular tools are required. They are required to edit the genome at the place of our own choice. And you should also understand the level of trouble and difficulty behind this exercise. So targeted interventions at a molecular level of DNA is like the thing the human species is trying to develop, because that is the only way when we might be able to control the functions of the genes. I can tell you, when the DNA was discovered in 1954, I guess, Watson and Crick, thanks to the Rosalind Franklin, they able to discover the structure of the DNA, and human race started feeling at that time they have discovered the code of life. They came to know that how nature and God has written the life. They came to know that how the life was written. And you know, in order to change something, the first step is that you need to understand the language. Like शेर कहने के लिए language एक primary जो है वो वजह होती. So we thought at that time that we know the language of life, at that language of the God, and now we can manipulate it as well. But it has been like 70 years or more than 70 years that we are struggling. We came to know about and we are like developing our knowledge about the language of life, and we are trying to develop better tools to edit the genome of human beings. And I can also tell you, and this is me, so you don't need to believe on that stuff. This is just something which I believe, that there is nobody is going to stop human race for that thing, because it is destined. It is like part of the human nature that life do not want to die. It will push the primary species, the best species, which is supposed to be humans, to keep finding the ways to help ourselves to exist forever. So we will keep trying to make our genome better and better and better. I don't know if this will going to be better or not, but what I do know is that there is no way that we can stop humans to manipulate human genome. That is, I believe, is a future, whether we like it or not. There are three big systems of molecular scissors or molecular tools are out there, and those molecular scissors and tools were used. Actually, they are developed one after the another. They are like stairs. They give birth to another one. The other one gives birth to another one. The most fascinating molecular scissors were zinc fingers at that time, because they were like new. They were more efficient than the traditional system, so they were accepted with a lot of applause at that time. I did join like 2010 for my masters, and I was studying that thing at that time. So like just 10, 11 years ago, it was just a highlight of research at that time, using zinc fingers to edit the genome at the place of your choice. This is called as zinc finger nucleases, or ZFN, zinc finger nucleases. I will explain you about them a little later. The next is that after that, in 2011 and 12, another more efficient versions of molecular scissors have been developed, and they are called as talons. Transcription activator like effector nucleases. And and please remember my words, mark my words, that if you want to stay in this area of research called targeted genome editing, this is like the most sacred thing you should know. Your professor will kick you out if you don't know these stairs. The first step is the zinc finger nucleases, then the talons. He will or she will wants you to know all this. And the last and not the least, the crown of everything is what we call as the CRISPR-Cas9 system. And everyone talks about CRISPR-Cas9. It's so lovely to speak about the CRISPR-Cas9 thing. But if you understand the journey, the development, it will be much easier for you to understand the CRISPR-Cas9 system. It's not a very difficult job, but the thought process, the evolution, the logic, the concept, that is the most important thing behind these discoveries. Understanding these things will going to expand your wisdom in a very different way. Okay, I have a question, and that question comes from Zunera. Onion genome size is about 15.9 GB, 5 times larger than human size. Doesn't necessarily imply exactly. More complex does it? Okay. Also, could you talk a bit about why onions are large? Oh, okay. Zunera ka question hai, before I go further, I think I have completed the stair stuff for you guys. Having a bigger genome should give us more complexity. That was the question. And if the size of the genome is related directly to the complexity or not. Otherwise, the onion should be more complex than us. In order to understand this question, you should understand the evolutionary procedure. The evolution starts from the same stem, the same place. And then it started separating. The plants go this way, animals go this way. And then, there are different branches, sub-branches, branches, some branches, and then things can go like further. I can go further, like evolution proceeds in this way. We know that. So all the animals and plants in their context, they try to evolve themselves according to their circumstances, according to their local address, according to their local situation and circumstances. If I ask you that the organism present here, please stay with me. If I ask you to the organism which is present here on this branch, whether this organism is related to the organism present here, the answer is yes. These two organisms are related. If I ask you whether this organism, number 1, is related to the organism present here, number 3, the answer is yes, they are related. Which one of them are more closely related with each other? Which one of them? 1 or 2? Let me see. 1 or 2 are closely related, or 1 or 3?
1 and 2. 1 and 2.
1 and 3, or 1 and 2. The 1 and 2 are supposed to be closely related because they have been separated in the evolutionary time scale, recently. Now think about the other side of the bar, which is like for the animals. If I reserve it for the animals, so whatever is present here, whatever the animal which is present on this side is very much different in the perspective or in comparison with any organism present on the plant side. So evolution separate it ways in two major. There was a major diversion at the very start of life, the autotroph and heterotroph, and then it leads to animals and plants. So whatever the animals were evolving, they get separated from the plants long time ago. Okay, their contexts are entirely different of evolution. That's the first thing. That is why there is no comparison of phenotype between two organisms if they are not closely related. And if they're plants and animals, I mean, like they're way, very different. And I can tell you, like, I think, make me correct if I'm wrong, or corrected by yourself if I'm wrong. Like maybe 60% of our genome is similar to banana. But does that look like 60% banana? I mean, I'm this no mean. So that's like it is more or less like that. Many of the enzymes which I am carrying, those enzymes are similar, like in banana as well. But since we have like got separated at a very, very early time, we evolved our genome in a very different way. So the complexity is not related with the amount of DNA somebody is carrying. It is related with the way and the context of evolution. And luckily, we have developed our whatever the genome we have left with in a better way. And unfortunately, the onions they fail to develop their. Oh, I can't tell you they are failed. Actually, they are successful as well in their own context. Onions are onions. I mean, one more thing you should know. That was also very exciting topic. I don't know why it is no more an exciting topic nowadays. I mean, earlier it was quite exciting. That topic was called as a reduced genome. I want you to know about these things because if you, if you able to talk to the other people in the future, you need to talk to the other professors and other people out there. You should have this concept very clear and right in your mind, that why people are doing all these things. That concept was called as reduced genome. Just try to browse this thing. And I have no idea recently how it was going right now. But earlier, like 10 years ago, it was very fascinating idea. The concept of reduced genome is that bacteria is carrying like 4.3 megabase pairs of genome in one chromosome. Most part is not producing anything, junk DNA, and also not involved in regulation. How about removing the extra unwanted stuff from the genome and create a bacteria with the less amount of DNA, but with the same function? So at that time, it was like there was a race to create the bacteria with a smaller and smaller and smaller genome. So they wanted to remove unnecessary parts from the bacterial genome. And I can tell you that was not, very much exciting nowadays, because in humans, we started understanding that the unwanted or non-coding part of the human genome is still actually able to control everything. And there is a lecture about control of gene expression at a molecular level. If you might be interested, you should browse and see. That was over there on my channel. That explains that how the 98% of the non-coding part of the genome, which is not involved in anything, is actually controlling the 2% of the coding region. Is it amazing? हम पहले उसको कहते थे junk DNA. That was junk DNA. That was not involved in anything. फजूल सी चीज है. But now we came to know the 98% of the junk DNA is actually controlling the 2% of the coding region. ये बिल्कुल ऐसे ही है जैसे space में ना. In the space we thought earlier there was nothing. The concept of nothingness is absolute, and there is nothing absolute in science. So हम समझते थे कि stars के दरमियान, between the stars, between the galaxies, there was nothing, and that nothing is called as the space. And later on we came to know that there was something out there, and that something like gravitational waves, and it was discovered later on experimentally as well. And there was dark matter between the stars and galaxies. So there is something between the stars and galaxy which we believe earlier there was nothing. And that something which exists between stars and galaxies is actually controlling the gravity between the stars and the galaxies. The reason why I'm telling you this, the things from the stars is that earlier we believe between the genes, whatever the stuff, whatever the DNA was present there was nothing, was not controlled, was junk. But now we know that the things which are present between the genes, those area was constituting like 98% of the genome, and that controls and regulate the expression of the 2% of the gene. So that's very, interesting and exciting. So let's start thinking about the first thing, and that first thing is about the zinc fingernail. But we